Hi class, this is your instructor, Skylar Huff, and we're now here in Chapter 12 with DNA, the carrier of genetic information. After the rediscovery of Mendel's principles in 1900, geneticists conducted experiments to learn how genes are arranged in chromosomes. They also want to know how they are transmitted from generation to generation. However, basic questions remained unanswered through most of the first half of the 20th century. What are genes made of? How do genes work? The studies of inheritance patterns described in Chapter 11 did not answer those questions, but they did provide a foundation of knowledge that enabled scientists to make predictions about the molecular or chemical nature of genes and how genes function. So with that, scientists generally agreed that genes would have to store information in a stable form that could be accurately copied and passed unchanged from generation to generation. It was also known that a gene could sometimes convert to a different and stable form that will be transmitted unchanged to future generations. These genetic mutations, these changes, were called and known as genetic mutations. As the science of genetics was developing, biochemists were working to correlate the known properties of genes with the nature of various biological molecules. What kind of molecule could store information? How could that information be retrieved and used to direct cell functions? What kind of molecule could be relatively stable but have the capacity to change, resulting in a mutation under certain conditions? As they learned more about the central role of proteins in virtually every aspect of cell structure and metabolism, some scientists considered these complex and diverse molecules the prime candidates for the genetic material. However, protein did not turn out to be the molecule that governs inheritance. So here in this chapter, we will discuss how researchers discovered that deoxyribonucleic acid, DNA, a nucleic acid, thought unremarkable, is the molecular basis of inheritance. So here we will explore the unique features of DNA, including its structure and replication. So that, of course, is what helped enable it to carry out its role. So let us begin now, class, in Chapter 12, Section 1, with evidence of DNA as the hereditary material. So as I go through this portion of the lecture, just keep in mind where we are. And some may say, but it's 2020 or 2022 or it's 2021. No, it's not. So let's go back into history, back in time. So we're going to go back, class, to the 1930s, 1940s. I'll begin now. So during the early 1940s and even into the 1930s, most geneticists paid little attention to DNA, convinced that the genetic material must be protein. Given the accumulating evidence that genes control production of proteins, meaning we'll get to that in the 13th chapter, class, it certainly seemed likely that genes themselves must also be proteins. Scientists knew proteins consisted of more than 20 different kinds of amino acids in many different combinations, which conferred unique properties on each type of protein. So given their complexity and diversity compared with other molecules, proteins seem to be the stuff which genes are made of. In contrast, scientists had established that DNA and other nucleic acids were made of only 4 nucleotides, and what was known about their arrangement made them relatively uninteresting to most researchers. So it is for this reason several early clues to the role of DNA were not widely noticed. So here we go with DNA being that transforming factor in bacteria. So please pay attention, class, and we're here with one of the very first experiments, and this is known as that transformation experiment. So one of these clues had its origin back in 1928 when Griffith, a British medical officer, made a curious observation concerning two strains of pneumococcus bacteria. So please see figure 12.1 in your text, found on page 255. I'll use this as our springboard here. So it was a smooth strain named for its formation of smooth colonies on a solid growth medium that exhibit virulence, and that just meant that it had the ability to cause disease and often death in its host. So when living cells of this strain were injected into mice, the animals contracted pneumonia and died. Not surprisingly, the injected animals survived if the cells were first killed with heat. Please follow me. A related rough strain of bacteria, which forms colonies with a rough surface, exhibited A virulence. That prefix A, of course, lets you all know that it has the inability to produce pathogenic effects. So mice injected with either living or heat-killed cells of this strain survived. So as you all can see here, class, the strain, class, that we noted, firstly, class, that were smooth, killed the mice. Nextly, class, we got to the rough strain, and of course, they were injected, or it was injected, and the mouse lived. And of course, I also mentioned that those heat-killed, smooth strain cells allowed the mouse class, or mice rather, to live. So it states, however, when Griffith injected mice with a mixture of heat-killed, virulent S cells, or smooth strain cells, and the live A-virulent, rough strain, or R cells, a high proportion of the mice died. And that's where we are here. So, with that being the case, Griffith then isolated some of the living smooth strain cells and the mice from the dead mice. So, because neither the heat-killed smooth strain nor the living rough strain could be converted to the living virulent form when injected by itself in the controlled experiments, it seemed that something in those heat-killed cells converted the A-virulent cells to that lethal form. This type of permanent genetic change, in which the properties of one strain of dead cells are conferred on a different strain of living cells, is called transformation. Scientists hypothesize that some chemical substance, a transforming principle or factor, was transferred from the dead bacteria to the living cells and caused transformation. So I'll say once more, this was one of the, I would say, first things that let us know, or let the scientific community know, that there is something that is being transformed. So just to make sure you got that, class, this is the very same definition just given. So next up, in 1944, we have Avery and MacLeod, including McCarthy, that, of course, carried out the experiment of Griffith, class. And just keep in mind, we're using the scientific method, class. Your results should be repeatable. So let's see if they were able to repeat those results of Griffith. So here, they chemically identified Griffith's transforming factor as DNA, deoxyribonucleic acid. They did so through a series of careful experiments in which they lysed, or split open, those smooth strain cells and separated the cell contents into several fractions: lipids, proteins, polysaccharides, and nucleic acids, being DNA and RNA. They tested each fraction to see if it could transform living rough strain cells into smooth strain cells. Experiments using lipids, polysaccharides, and proteins did not cause transformation. However, class, when Avery treated living rough strain cells with the nucleic acids extracted from the smooth strain cells, the rough strain cells were transformed into the smooth, virulent S cells. Although today scientists consider these results to be the first definitive demonstration that DNA is genetic material, not all scientists of the time were convinced. And I can assure you, class, that this let us know that DNA is indeed genetic material. So let us now continue on. And just before we get to where we're going, let me just stop once more and state that many of the scientists of that time thought that the findings might apply only to bacteria and not have any relevance for the genetics of eukaryotes. So during the next few years, new evidence began to accumulate, and it was that the haploid nuclei of pollen grains and even gametes of sperm contain only half the amount of DNA found in diploid somatic cells of the same species. Because scientists generally accepted that genes are located on chromosomes, these findings correlating DNA content with chromosome number provided strong circumstantial evidence for DNA's importance in eukaryotic inheritance. So let's get now to 1952. So just keep in mind here where we are in time. So this is now the 50s. We have two geneticists here, Hershey and Chase, and just keep in mind that science can be done, class, by women. In the case of the class of Martha Chase. So what Hershey and Chase did was perform a series of elegant experiments on the reproduction of viruses that infect bacteria. They are known as bacteriophages, or just phages. We'll get to this, class. We'll learn about bacteria here shortly. When they planned their experiments, they knew that phages reproduced inside bacterial cell, eventually causing the cell to break open and release large numbers of new viruses. Because electron microscopic studies had shown that only part of an infecting phage actually enters the cell, they reasoned that the genetic material should be included in that portion. As shown in figure 12.3, and please look carefully, figure 12.3, class, is in your textbook on page 256. That class will show you everything that I'm about to get to now. So let's begin. They labeled the viral protein of one sample of the phages with 35S, and I'm referring, class, to sulfur. And if you can think back, class, let's see here. Where is my figure I wanted to show you? Oh, where can you be? I thought I added that figure to your presentation, and apparently Huff did not. I apologize. So again, I say, in your text, class, please ensure that you go to page 256 and see figure 12.3. This class will help you with what I'm doing right now. So what I'm getting at is that radioactive isotope of sulfur is there. So this takes you all the way back, class, to, I would like to say, chapter 2. In chapter 2, you learned that radioactive isotopes are used in experiments as tracers. So it's used now, class, to trace. Get back to where I was. So they also, class, labeled the viral DNA of a second sample with 32P, class. That is the phosphorus portion, as you all see there. So they label, class, the radioactive sulfur as well as the radioactive phosphorus. Keep in mind. So the protein, class, was labeled by way of sulfur because, of course, the sulfur is within the protein itself, as opposed to, class, them labeling the radioactive phosphorus. And you all should definitely know by now, class, about the structure of nucleic acids, particularly, class, that phosphate backbone. Hence, they labeled, class, the radioactive phosphorus. I'll continue. So, in chapter 3, class, you all, I hope, learned that sulfur is a part of amino acids, particularly in this case, class, cysteine. cysteine, as well as methionine. And methionine is a very important nucleic acid. I'll continue. So I've already mentioned that nucleic acids contain the phosphate groups that make up the phosphate backbone. So the phages in each sample attached to bacteria. I guess I'll show figure now. I'll now continue. So the researchers shook the phages off by agitating the sample in a blender. They then centrifuged the samples. And the sample in which the protein was labeled with, of course, the sulfur, they subsequently found radioactivity in the supernatant class, indicating that the protein had not entered the cells. In the sample in which they had labeled DNA radioactively by way of that phosphorus, they found radioactivity associated with the bacterial cells. In other words, that class was found in the pellet. DNA had actually entered the cells. Hershey and Chase concluded that phages inject their DNA into bacterial cells, leaving most of their protein on the outside. This finding emphasized the significance of DNA in viral reproduction, and many scientists saw it as an important demonstration of DNA's role as the hereditary material. So class, upon completion of this experiment, it was stated, class, that DNA is genetic material. So, getting into the structure here now, class, I'll just be blunt. Scientists did not generally accept DNA as the genetic material until 1953. And here, class, we get to two other scientists. These scientists, class, are known as Watson and Crick. So let's get here now. Watson and Crick, class, worked in England, and they proposed a model for the structure of DNA, which had that extraordinary explanatory power. And in the words, if I'll say it this way, the story of how DNA structure was figured out, class, is one of the most remarkable chapters in the history of modern biology. So as you will see in the following discussion, class, scientists already knew a great deal about DNA's physical and chemical properties when Watson and Crick became interested in the problem. In fact, Watson and Crick did not conduct any experiments or gather any new data to get to this structure of DNA, class. Nothing new was done. But yet and still, class, they are, class, in, I would say, every biology textbook. Let us continue. Their important contribution was to integrate all of the available information into a model that demonstrated how the molecule can carry both information for making proteins as well as serve as its own template, as its own pattern, or as its own guide for its duplication. So on page 257, just keep in mind, class, there is a timeline of selected historical DNA discoveries. And for those of you who love history, I would say please check that out, because I'm going through pretty much every part and piece of that, class, today in this lecture. So what I'll get to here is another thing I'm missing. Oh boy, let's stop here now. This is where I wanted to go, class. So it mentions here, from 1951 to 1953, class, here is yet another scientist. And this scientist is named Franklin. And I have to say it again, class, it wasn't just Martha Chase. It was also, class, Rosalind Franklin, another woman here in science. So I must encourage you, women, young ladies, females, please get involved in science. There should be, of course, more women in science. There should be more minorities. I'm not just referring to women as a minority or females as a minority. I'm also referring to, of course, those, of course, who are black and brown. There should be, of course, more people of African descent here in biology. And, of course, more people, class, of, I would say, Hispanic descent. So please just know, class, that there is space here in science for you. And of course, there should be more representation, class, of minorities. Let us now continue with the lecture. So here in this case, class, Franklin used X-ray diffraction. And what she did in using X-ray diffraction, if you look with me, class, on page 258, this is key information about how the structure of DNA came from X-ray diffraction studies on crystals of purified DNA. And she carried these out, class, at King's College. So X-ray diffraction, as it is, I'll just go here now, showing you how this works. It's a powerful method for determining the 3D structure of a molecule, and it can determine the distances between the atoms of molecules arranged in that regular repeating crystalline structure. And as I say that, see figure 12.6 on page 259. X-rays have such short wavelengths that they can be scattered by the electrons surrounding atoms in a molecule. Atoms with dense electron clouds, such as phosphorus and oxygen, tend to deflect electrons more strongly than atoms with lower atomic numbers. So exposing a crystal to an intense beam of X-rays causes the regular arrangement of atoms to diffract or scatter the X-rays in specific ways. So the pattern of diffracted X-rays appears on photographic film as dark spots, as you all see there. Mathematical analysis of the pattern and distances between the spots yields precise distances between atoms and their orientation within the molecules. Franklin had already produced X-ray crystallographic films of DNA patterns when Watson and Crick began to pursue the problem of DNA structure. Her pictures, yes, I just said her pictures, clearly showed that DNA has a type of helical structure and three major types of regular repeating patterns in the molecule. And the dimensions were 0.34 nanometers, 3.4 nanometers, and 2 nanometers. This was all evident. So Franklin and her colleague Wilkins had inferred from these patterns that nucleotide bases, which are flat molecules, are stacked like rungs of a ladder. So to get to where I'm going with this, class, and to make sure nothing is left out, what Franklin did actually is what enabled Watson and Crick to get to what they did. In other words, the work of Franklin, class, occurred in 1952. The work of Watson and Crick occurred in 1953, as well as other work they did, class, occurring not long thereafter. I'll put it this way. As Watson and Crick worked, it was shown to them, unbeknownst to Franklin, what she was doing, meaning a picture. The very picture you all see here. It was shown to them, her work. And from them seeing this, with that kind of, I guess I'll use this X-shaped pattern here, Watson and Crick, class, was able to create the first three-dimensional model of DNA. And they did that without her permission. And originally, she gained, I guess you would call, class, no credit for her work initially. But ultimately, class, Franklin was given the appropriate credit for her work that enabled, class, Watson and Crick to create that first three-dimensional model of DNA being a double helix. So I say this to say, just make sure, class, that you understand that whatever you do, class, will be noticed, even if it's not noticed, of course, thereafter when you do it. The work class of this woman is why we are where we are today. And we could not have gotten there, class, I would say, without the work of Rosalind Franklin. Let us now continue. So continuing with the DNA structure class, just keep in mind that the building block of DNA is known as the nucleotide. It has three pieces, class. It has that pentose sugar, the phosphate backbone, class, and one of four nitrogenous bases. Those nitrogenous bases, class, are always going to be attached to that 1' carbon of the sugar, and the phosphate group is always going to be attached to that 5' carbon. The four nitrogenous bases of DNA, class, are as follows. They are adenine, guanine, thymine, and cytosine. Class, these are all things you all should know quite well, so I will not, class, spend much time here. I'll now continue. So the nucleotides are linked by covalent bonds to form the alternating class sugar, phosphate, backbone. Sugar, phosphate, backbone. So that 3' carbon of one sugar is bonded to the 5' phosphate of the adjacent sugar to form that 3 to 5' phosphodiester linkage. I repeat, they form a 3' to 5' phosphodiester linkage. And the result is a polymer of indefinite length with nucleotides linked in any order. So scientists now know that most DNA molecules found in cells are millions of bases long. So with that being the case, class, just know that a single polynucleotide chain is directional, and no matter how long the chain may be, one end is 5'. It has a 5' carbon attached to a phosphate on the other end, and that's that 3' end, which of course has a 3' carbon attached to a hydroxyl group. So as I say this, to not take up too much time in this lecture, class, let's get to a bit of work here to draw. Let's just draw it this way. So here it is. So we have this there being that phosphate group, and then we now have, class, the 5 carbon sugar. And then, of course, we do have here, class, that nitrogen base. So in this case, I'll put a T, class. The T is for thymine or thymine. And what I'll also now do, class, is I'll draw the other, class. And here we go. In drawing the other, I'm going to add, since I have thymine, I'm going to add my adenine, and it's going to be connected. Let's make our connection, Huff. I'll connect it here with, I'll go with this here. So those are two hydrogen bonds. I'll do, class, more of that a bit later. I'm not quite done. So now I'll draw yet another box. And this, class, is going to be adenine. And I drew this near the top, which was such a mistake. But oh well. So now that I've drawn that, I'll now get to... Yeah, I made such a mistake drawing this near the top, class. Like this. And then I'll go there and there and there. I don't know how on earth this thing keeps doing that. Sorry about that, class. And there we go. So I'll now get to a portion I wanted to get to moments ago. And I'll number my carbons. 1, 2, 3, 4, 5. And here is 1, 2, 3, 4, and 5. So what I've done for you, class, is I've gotten to, I would say, what you need to get. In other words, we have together here two nucleotides. As we have those two nucleotides here, class, this exhibits that here on the left, this is 5' to 3' going in that direction, and that, class, is 3 to 5' going in, I guess you would say, that direction. Or in other words, class, what I'm showing you here is DNA is anti-parallel. Of course, you have your two nitrogenous bases here We have class here the sugar deoxyribose. We have the sugar deoxyribose. And of course, we have our phosphate backbone being created there. The phosphate backbone being created there. So I only drew one of which on each side, but this class definitely shows you 5' to 3', 3' to 5' class. This is DNA. So let me get back to where I just was. So this shows you, class, more or less what I just drew. But of course here, you're not necessarily seeing, class, every part and piece. So let's get to, of course, how we have this phosphodiester linkage. Here, of course, I already mentioned the sugar and nitrogen base there. Let's continue. So I'm now getting to Chargaff's rule. So in 1949, Chargaff and others found the relationship among DNA bases that turned out to be an important clue in DNA structure. So the rule, class, is as follows. A equals T and G equals C. Meaning, he found, class, in double-stranded DNA molecules, the number of purines equals the number of pyrimidines. So I'll say that and pause for just a moment to ensure it sets in. And I guess to ensure it's not forgotten, class, when I mention, of course, the purines and pyrimidines, just look closely, class. For instance, let me change my color, class, to this. Here we go. So I guess I'll start off with the purines. Purines. So our purines, for those that may have not remembered from chapter 3, are adenine and guanine. A as well as G. These, class, are purines. Purines. And then the pyrimidines, which of course is spelled with a Y, pyrimidines, they are cytosine and thymine. And then, of course, I'll put a semicolon here, class, and put this in parentheses because it does indeed, class, include uracil. And I did that because we find uracil, class, in RNA, which we're not quite there yet. In other words, class, as you've learned before, one purine, class, will always be paired with one pyrimidine. Keep in mind, class, that's that. I'll now continue. So, using what Chargaff and others came up with, class, he just found out and saw that the percentage of bases were, of course, an approximate 1-to-1 ratio. And, of course, you see that now, class, with the adenine, class, and thymine here, including, class, guanine and cytosine. So there you have it. And this is not, class, restricted just to us being humans. We also see this, class, in corn, the plant, ZMAs, even, class, in yeasts, the fungus, Saccharomyces cerevisiae, and, of course, in Escherichia coli, E. coli. So I've already gotten here, class, to the contribution of Franklin. So I'll now just continue on, class, with where we've left off. So to make sure you've gotten it, class, it states here that to fit that X-ray data to the two chains, of course, they must run anti-parallel. And I showed you that, class, moments ago. So I also wrote this out for you. So if you look, class, at figure 12.9 in your textbook on page 260, what you would see, class, is that adenine and thymine, they're joined, class, by two hydrogen bonds. Whereas guanine and cytosine are joined, class, by 1, 2, 3 hydrogen bonds. So keep that in mind, class, as far as structure. So structurally, we've already gotten, class, to complementary-based pairing, but I must do this yet again because on your test you will see this. So I'll use what you're being shown for example, and I can't stress enough, class. The sequence of nucleotides in one chain definitely dictates the complementary sequence of nucleotides in the other. So let's just go with this 5' side, and I'll write down, class, the 3' side. This should make very well, class, sense. If it does not, you should tell me. So I'm writing what's on your screen, class. C, G, A, T, G. So we have that sequence of 5 nucleotides by way of those nitrogenous bases. So that complementary strand, class, would be, of course, going in that 3'. Well, let me go ahead and go a little bit different here. A little different. That's just to make sure this is something easily seen. So the complementary strand, class, would be in that 3' to 5' direction, because yes, this is a double helix. And keep in mind, class, that it is anti-parallel. Anti-parallel. So, of course, C, class, pairs with what? Definitely G. So, of course, this G, called guanine, will yet again, class, be paired with cytosine. Adenine, class, pairs with thymine. Thymine, class, pairs with adenine. And this last guanine, class, will be paired with cytosine. So this is the very same, class, that you'll be tasked with doing on your lecture test, and this shows you how it works. I'm continuing now. And now you see, class, by way of hydrogen bonding. I wasn't quite sure, but this is definitely, class, figure 12.9 from your text from page 260, with 2 hydrogen bonds, class, here between adenine and thymine, and 1, 2, 3 hydrogen bonds, class, here between the guanine and cytosine. I'll now continue. So we now get to DNA replication. You'll find this, class, on your test. But you haven't quite gotten to the process of it. But of course, on the test, you'll be asked to describe the process of replication, i.e. your test homework. So here we are. So the DNA molecule suggests a precise replication sequence of nucleotides. I don't know what in the world that A is doing there, but of nucleotides. So the immediate and apparent distinctive features that Watson and Crick's model made, it seemed plausible that DNA is genetic material. So we have already mentioned the sequence of bases in DNA that carry the coded information. So the model also suggested a way in which the sequence of nucleotides in DNA could be precisely copied, and that process is known as DNA replication. So as we get to that here, class, just make sure you remember that each strand of DNA molecule serves as a template strand for synthesizing the opposite strand. And to not necessarily get ahead of myself here, but I'm on page 261. And as I get here, it just says the connection between DNA replication and the behavior of chromosomes in mitosis was obvious to Watson and Crick. So the chromosome, it becomes duplicated so that it consists of 2 identical sister chromatids that later separate at anaphase. So that genetic material must be precisely duplicated and distributed to those daughter cells. So they noted in a classic and now famous understatement at the end of their first brief paper. Check this out. It has not escaped our notice that specific pairing have postulated immediately suggest a possible copying mechanism for the genetic material. In other words, class, their model suggested that because the nucleotides pair with each other in a complementary fashion, that each strand of DNA could serve as a template for synthesizing the strand. So it would be necessary for the hydrogen bonds that I just showed you, class, to break, and then the 2 chains to separate. And just keep in mind, class, the hydrogen bonds are relatively weak. So each strand of the double helix could then pair with a new complementary nucleotide to replace its other missing partner. So the result would be 2 DNA double helices, each, class, identical to the original one, and consisting of 1 original strand and 1 parent molecule, and newly synthesized complementary strand. So this type of copying, class, is called semi-conservative replication. Write that down. So with that being the case, there were alternative methods, and one of which class is conservative replication, and the other is dispersive replication. So Meselson and Stahl verified the mechanism of semi-conservative replication. So what we're seeing here, class, firstly is semi-conservative replication here. Semi-conservative replication is there at A. At B, class, that is conservative replication. And then here at C, class, is dispersive. It is the dispersive replication. So I know those pictures may not look like much, so let us continue on, class, so I can make this make a lot more sense. So what did Meselson and Stahl do? Well, they conducted an experiment. Yes, this is biology, so of course it is rife, class, with experiments. Experiments galore. So although the semi-conservative replication mechanism suggested by Watson and Crick and was and is a simple and compelling model, experimental proof was needed, class, to establish the fact that DNA in fact replicates in that manner. So with this, let's get to how it happens. So you can see this, class, explained, beginning here on page 262 and continuing on, class, to page 263. So what happened is they hypothesized 3 different hypotheses of DNA replication, class, each of which, of course, predicts the arrangement of new and old synthesized DNA after 1 or 2 generations according to each hypothesis. So there they are, class, the semi-conservative model, the conservative model, and the dispersive model. So what they did, class, Meselson and Stahl, grew bacteria, here E. coli, in heavy nitrogen growth medium for many generations so that all the DNA strands would be heavy. So they then, class, transferred some of the cells into light nitrogen medium so that the newly synthesized strands would be light. From that, they isolated the DNA from the bacterial cells after 20 minutes, that is 1 generation, and after 40 minutes from 2 generations. And then they centrifuged it, class, to separate the DNA into bands based on density. So here we go. They used a density gradient centrifugation. And when I mentioned this density gradient centrifugation, class, they used cesium chloride. And I guess you may have heard of cesium or cesium chloride. But they came up with this density gradient centrifugation. And what they did, class, is in the density gradient centrifugation, it was the concentration of the cesium chloride and its highest, class, at the bottom. The concentration is highest here at the bottom. The concentration, class, is also, of course, lowest at the top. And then, of course, the DNA molecules move to positions where their density equals that of the cesium chloride solution in which they are centrifuged. So, class, I hope you all are getting what I'm going to. So at the bottom, of course, we have the heavy DNA. At the bottom was the heavy DNA. That 1 cell generation, class, after that transfer was here, class, in the middle. And then, of course, the light DNA was there at the top. From this, class, and based on the observed density of the DNA molecules in each generation, Meselson and Stahl concluded that semi-conservative model of replication accurately depicts the mechanism of DNA replication. So the location of those molecules within the centrifuge tube were, of course, determined by ultraviolet optics, and the solutions, of course, absorbed strongly at 260 nanometers. So it was this, class, that, of course, let the entire community know that as it happens, class, you have every time, class, one old strand of DNA and one newly synthesized strand of DNA, all by way of replication. Thanks a lot, Meselson-Stahl. So amazing. So this class, being that semi-conservative replication, provided the mechanism for genetic changes and their transmission to succeeding generations. So what I'm getting to here, class, is it's that the DNA that could be copied, class, in this semi-conservative mechanism is what led to that explanation, class, of a third essential characteristics of DNA, meaning its ability to mutate. So it was long known that mutations, or those genetic changes, class, could arise in genes and then be transmitted to succeeding generations. And of course, this was even shown, class, in Drosophila melanogaster, meaning a mutation in the fruit fly that produces those vestigial wings. So with this, those mutations, meaning the changes in the sequence of bases in DNA, which are produced in a new sequence of complementary bases in that next replication cycle. So I'll just put it this way, class. If there's a DNA that, if there is a mutation, class, in the DNA that is there, that very same mutation, class, will show up yet again, class, there. So this is why, class, it has been stated that semi-conservative replication explains, class, how mutations, class, can be perpetuated. And it may be obvious, but I say it's because it is in the DNA. Let's continue. So DNA replication requires protein machinery. With this, class, I'm going to get to the process of replication. So keep in mind that this is what you'll be tasked with explaining on your test. So DNA replication starts at origins of replication, where small sections of the double helix unwind. And as I say, small sections of the double helix unwind. Where am I trying to go? I'm trying to go here. So this is that unwinding, class, I'm mentioning here. So it unwinds. So as it unwinds, the reason being so, class, is because of DNA helicase. I repeat, helicase, class, is what is there. And as helicase is there, it actually unzips that double helix to create those two replication forks. So that is helicase. And now, class, we have those two replication forks, all because of DNA helicase. So now that we have those two replication forks, let's get to the process, class, of replication and how it occurs. All right. So as helicase unzips those two strands that make the double helix, we now, class, have the origin for replication, class, here. And of course, those hydrogen bonds, which were once there to hold together those nitrogen bases, of course, are no longer there. Thanks a lot to the two, class, between guanine and cytosine. I'm sorry, the three between guanine and cytosine, and those two, class, between adenine and thymine. So what helicase does is it continues to travel, class, along that helix to continue opening the double helix, class, almost like a zipper. I know you all have used a zipper on a jacket or a coat or on whatever, class, has a zipper, and that's what helicase does. It unzips, it unwinds that double helix. So what's next, class, is once helicase has separated those strands, it's called single strand binding proteins that bind to DNA strands and stabilize them. So they're called SSB proteins. So that prevents the double helix from reforming until the strands are replicated. So the SSB, or those single strand binding proteins, they also prevent the hydrolysis of a single strand region by other enzymes, meaning it keeps those enzymes from breaking down the what is now that arm, or of course that replication fork. Because nucleases, class, we'll get to later, could of course very well do such a thing. So it mentions, class, here a few more proteins, a few more enzymes. So we have DNA polymerase. So what DNA polymerase does, and you may also hear some textbooks, class, call it DNA polymerase 3, depending upon who is teaching it or teaching you. So what DNA polymerase does is it comes along and it adds those nucleotides. It adds those nucleotides, and it adds those nucleotides, class, of course, based on that template strand. In addition, class, to adding those nucleotides, what DNA polymerase does is, of course, checks for errors. It checks for errors. Let's continue on. As I mentioned, class, DNA polymerase, it links those successive nucleotide subunits, meaning as they become linked, those two phosphates are removed. So the DNA polymerase, as I mentioned, it adds nucleotides only to that 3' end of what is always there. So I believe this very well be a good time for me to just begin to draw. Let's get to drawing. So as we are here, let's go ahead and I just start like this. So that class is a template strand. This class is yet again another template there. And I'm going to now add bases. And these bases, class, are being added, I would say, so arbitrarily. So I'll make this side class 5', 2, 3'. This side will most definitely be class 3', 2, 5'. So I already mentioned, class, the role of our unzipper class being DNA helicase. So since we already have helicase, which has come along, and unzipped class, what was once here, and I kind of just draw it here. Hi, that's helicase. And I have a feeling I just run out of real estate here. Whatever real estate is. Yeah, helicase. That's HELICASE, helicase. So let me add my nitrogenous bases here. And I told you this is so arbitrary. So A, T, C, C, G. Now what's class here on my 3' to 5' side is not arbitrary at all, meaning they have to match. So this is T as in Tom. This is T as in Tom class, A as in apple, G as in go, G as in go, and then C as in cat. So now, class, we have our I guess you say the workings of what's needed. So I'll now, class, get to yet another set of enzymes. So we now have our RNA primer class and primase. So the RNA primer class as well as primase, what they do is they're their class to start the process of replication. So the primers and primase. So what we'll do here now is continue in that direction. Hey, where's my hot diggity? I'll have to draw this way, class. We will now, class, go in this direction with replication. So what I'm saying is this. It's all by way of DNA primase. I mentioned primase. Well, primase, class, is what makes those short RNA primers. And we must have this to start replication, class, here on our leading strand. So DNA polymerase comes along, class, and adds a C. It adds a C. It adds a G. It adds another class G. And just keep in mind, as this is happening, this class happens in the 5' to 3' direction. Notice, 5 to 3. And I'll call that yellow strand I just created, class, with those purple nucleotides, the lead or leading strand. It, class, happens, being replication on the lead strand, it happens in a continuous way. It's continuous on that lead strand. So on the opposite end of things, class, yet and still in the opposite direction now. So I'm going in this direction. So there's one. And it started all right there, class, yet and still with our primase. Yet and still, class, with primase and our RNA primers right there. And it starts again, yes, I said again, it starts again, class, right here. Still, class, going that direction. And in other words, class, I'm using red, so the colors are different. So on this red strand, still, class, in the 5' to 3' direction. Still, yet again, here in the 5' to 3' direction. Because I'm saying this, and I'll put it this way. Replication, class, will always proceed in the 5' to 3' direction. And not just replication, but also class transcription and even translation. We haven't gotten each of those yet, but I must make that plain. So I'll make these, I guess I'll go with a white. So there is, since I have guanine, this will be cytosine. Since I have guanine, this too will be cytosine. And since I have cytosine, this will be class guanine. So what I've created, class, here on this right-hand side are known as wow, that looks horrible as far as penmanship, but oh well. They're known as Okazaki fragments. Those are Okazaki fragments, and they are their class on what is known as the lagging strand. And I say it's the lagging strand because here, class, replication happens in a discontinuous fashion on that lagging strand. So there are 2 Okazaki fragments there. So I just put a 2 in parentheses, being, of course, there and, of course, there. So their class, replication proceeds more slowly. It lags behind our yellow lead strand. So just don't forget, I said on the lead strand, replication continues, class, continuously and smoothly, as opposed to that lagging strand, because it always grows away from that replication fork, class, which I put to start the process. And of course, it just lags behind. So with this, I guess I'll leave it here for just a moment, and I'll continue on, class, where we left off. So where I left off was, I guess I showed this, that's here. And then, of course, I mentioned the primers, class. They're synthesized where replication begins. And then here we are. So this just shows, class, that simplified view of what I just drew for you all. So yet again, class, DNA synthesis by way of replication proceeds only in that 5' to 3' direction. Only in the 5' to 3' direction. So DNA replication always begins at an origin, and both strands will, of course, go through the process of replication at the same time. So it's, of course, happening on each side, class, simultaneously, just in opposite directions. So the position of that replication fork is constantly moving, class, through the process. So going through this, class, what's here in words, I've already explained, class. So it's nothing that I guess you would say you haven't gotten yet. So the lead strand or leading strand, class, as I mentioned earlier, proceeds, class, continuously in that 5' to 3' direction, and the lagging strand, class, proceeds discontinuously, lags behind, class, growing away from the replication fork. All right, so what I'll get to now, class, is it states that fragments are joined by DNA ligase. What fragments? Okazaki fragments. So another enzyme, class, yes, enzymes galore. It's ligase, DNA ligase, that stitches together what are those Okazaki fragments, as I drew, class, moments ago in white, or encircled in white. So that ensures, class, that is a continuous strand of DNA as a process has occurred. So this, class, shows, by and large, the same thing I just drew for you. So you see here, class, still going in that 5' to 3' direction. You're seeing, class, that lead strand, and then you're seeing, class, coming away from that replication fork, the lagging strand by way of those 1, 2 Okazaki fragments, which of course will eventually be stitched together by DNA ligase. Thanks a lot, all these enzymes involved. So just keep in mind, as I mentioned moments ago, class, it happens in both directions, it being, class, DNA replication. All right, so during replication, class, DNA polymerases is already mentioned. Not only adds those nucleotides to create the strand of DNA, but DNA polymerase is also here, class, to check for errors, meaning it proofreads each added nucleotide to ensure, class, we have that exact copy. However, class, in the event, I repeat, in the event that there is an error that is found, class, DNA polymerase immediately removes that incorrect nucleotide and then inserts the correct nucleotide. So uncorrected errors, class, by DNA polymerase are repaired, of course, by other mechanisms. So other, of course, repair mechanisms include mismatch repair. And this is when you have special enzymes that recognize incorrectly paired nucleotides and then remove them. So the DNA polymerase then, of course, will fill in those missing nucleotides. Secondly, class is nucleotide excision repair. So this is very important, class, meaning that it's commonly used to repair DNA lesions or deformed DNA. Typically, class, this is what is caused by the sun's ultraviolet rays. Please, class, wear sunscreen. Living around, of course, I would say the southeastern United States, the rays here, class, are particularly harsh. And of course, this could, of course, happen, class, by way of some chemical. But I'll keep on going. So there are 3 enzymes, class, involved in nucleotide excision repair. So what happens is nuclease, it comes along, class, and cuts. It's like scissors. It cuts out the damaged DNA. Then yet again, DNA polymerase adds the correct nucleotide, and then just as you all learned moments ago, DNA ligase stitches back. It closes in those breaks to ensure, class, we have that DNA molecule. So in the event, class, that an individual has the disease, class, xeroderma pigmentosa, they have an inherited defect in nucleotide excision repair. So that enzyme, of course, is that affected part. So affected individuals may develop skin cancers at an early age because DNA lesions caused by UV rays, class, cannot be repaired. So this class is showing the process. We have the DNA lesion, class. The nuclease is there to, of course, remove. Upon being removed, class, DNA polymerase comes in as a nucleotide. DNA ligase, class, of course, comes in to stitch. So to end the chapter, class, and we're just about there, we get to telomeres. So telomeres are what cap off eukaryotic chromosome ends. I say again, telomeres, class, are what come in and cap off eukaryotic chromosome ends, and they do this, class, to protect. They do this to protect those ends of our chromosomes. And it does this because they begin to shorten. They begin to shorten, class, during replication. So those telomeres, if you're wondering, okay, Huff, what on earth is a telomere? Well, a telomere, class, is, I would say, nothing more than repeating non-coding guanine-rich DNA sequences. So they are those non-coding guanine-rich sequences that repeat many, 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 many times. So to continue, class, there, I'll just say that even in human sperm cells and egg cells, the sequence, class, from 5' to 3' is TTAGGG. I said TTAGGG, and that's from 5' to 3'. But their, class, is repeated more than 1,000 times on the end of each chromosome. Therefore, class, although a small amount of telomeric DNA fails to replicate each time the cell divides, a cell, class, may divide many times before it starts to lose any essential genetic information. So I hope you now, class, very well understand the importance of telomeres as they cap off those ends. So now let's get to an enzyme, class, called polymerase. So telomerase is a DNA replication enzyme that can lengthen the telomeric DNA by adding repetitive nucleotide sequences to the end of our eukaryotic chromosomes. So now that you get why that's important, there was a Nobel laureate, class, by the name of course, Blackburn, and the other of which being Grider. Class, I'll tell you, these women are doing science and doing science well. I say keep it up. And if you don't know if you want to do it, please contact me. I'll definitely support your class in any and every way I can, because yet again, more minorities are needed in science. So what happens is, typically, class, active cells, of course, that divide an unlimited number of times, meaning this enzyme was discovered in those types of cells, and this is included in protozoa and other unicellular eukaryotes. And, class, the biggie here is found, class, in most types of cancer cells. That's the biggie. I'll make it make sense. In humans and other mammals, active telomerase is usually present in germline cells, which of course give rise to egg and sperm. And even those rapidly dividing cells, such as our blood cells, intestinal lining cells, and skin cells, but not, class, in most adult tissues. To make it make sense, class, just keep in mind that typically found in those cells that divide an unlimited number of times, and most cancer cells. So what I'm getting here now to is that the shortening of telomeres may lead to cell death, class, programmed cell death known as apoptosis. And to give you a kind of, I guess you say, a view into what I'm referring to, class, is that a 70-year-old class, a 70-year-old has cells, just like we have cells. But of course, their cells may only divide class an approximate 20 to 30 times. However, class, an infant, which of course I have an infant at home, at least right now I do. An infant class, of course, has cells. Yes, of course they have cells. And their cells, class, divide an approximate 80 to 90 times. So I'm telling you this, class, to say just one more. Cancer cells have an unlimited number of divisions. So just keep in mind, class, that there is some correlation between the telomeres and telomerase, especially, class, when it comes to, of course, cancer. In other words, most cancer cells, including human cancers of the breast, the lung, the colon, the prostate, and the pancreas, have active telomerase, and that is what allows them to, of course, maintain telomere length and possibly resist, class, programmed cell death. So, of course, research, class, is being done, of course, to target, meaning to get these cancer drugs that target telomerase enzyme as an anti-cancer therapy. Class, this has been your instructor, Skylar Huff. This has been Chapter 12. Study well, class. Prepare for class. And if you have any questions, come by my office. Email me. See me before lab or even after lab. Thank you all for listening. Take care of yourselves.